Hi, welcome to my YouTube channel Felshestein. My name is Andrea and today we're going to have a look on a section for a spleen, which is the biggest peripheral lymphoid organ in the human body. Spleen is located in the abdominal cavity, right under the left part of the diaphragm. Spleen is unpalpable in normal physiological state, and enlargement of the spleen can indicate many pathological processes. Spleen has a capsule on its surface, which is made of dense, irregular connective tissue. It's wider in the hilum of the spleen, whereas the entry of an artery and nerve, vein and efferent lymphatics are coming out. You cannot find hilum on this slide. Section has been made on the periphery of the spleen. Capsule contains several myofibroblasts. They're just fibroblasts with the ability to contract. Some animals do have many myofibroblasts in the capsule of their spleen. It allows them to contract the spleen if necessary. Trabecules extend from this capsule and divide the spleen into compartments. Imagine trabecules as columns. We already saw them in the second part about the lymph node. Trabecular arteries come deeper through the trabecules. They further divide in the spleen. You can also see trabecular vein here. Spleen consists of two major parts, white pulp and red pulp. White pulp represents 20% of all organ and it's typical lymphatic tissue. Supporting structure is made of reticular connective tissue. You can also find B lymphocyte, T lymphocyte, dendritic cells, which are antigen-presenting cells, and macrophages between reticular cells and reticular fibers. White pulp is attached to the blood vessels. Already mentioned trabecular arteries that originate from splenic artery branch into several arteries called central arteries. Although they are called central, they do not pass through the center of the white pulp. We differentiate several parts in the white pulp. The one that is located near the central artery, called periarterial lymphoid sheath, and peripheral white pulp that is composed of the marginal zone of a white pulp and the lymphatic follicles. Periarterial lymphoid sheath is time-dependent. It really means that this sheath is expecting a delivery of T lymphocytes from the thymus. Lymphatic follicles located in the peripheral white pulp contains especially B lymphocytes. Their structure is the same with the follicle you saw in the lymph node. For a reminder, follicle consists of the germinal center, mental zone, and marginal zone. B lymphocytes by several processes differentiate into memory cells and plasma cells that secrete immunoglobulins. Besides the follicles, the peripheral white pulp contains also the marginal zone, that is at the interface between the white pulp and red pulp. Marginal zone contains blood sinuses where arterioles from central artery drain. There are fewer lymphocytes but numerous dendritic cells picking up antigens and macrophages which remove all the damaged cells and foreign particles. So, in general, two marginal zones can be distinguished in the spleen, the one in the follicle and the second one located in the peripheral white pulp, which plays a major role in immunity. Red pulp consists of cords of Billroth and venous sinuses. Cords of Billroth are made of reticular connective tissue, that's basically reticular cells and fibers, B lymphocytes, plasma cells, which are effector cells of the B lymphocytes, macrophages, red blood cells or erythrocytes. We can also find here granulocytes, monocytes, and platelets. These cords anastomose with each other and create network around venous sinuses. Venous sinuses are lined with endothelium. Don't mislead them with lymphatic sinuses, which can be found in the lymph node. They aren't the same structure. Venous sinuses are specialized vessels, and it's not a mistake to call them sinusoids. There are spaces between the endothelial cells that allow the exchange of nutrients and cells between the blood and pulp in the spleen. So, let's repeat it again. Blood supply for the spleen is from splenic artery, which enters the spleen in the hilum. It's branching into smaller trabecular arteries, which are located in the trabecules of the spleen, of course. Trabecular arteries branch into central arteries coated with a white pulp that emit branches for its supply. All branches of trabecular arteries are very small, and strictly morphologically they are arterioles. After leaving the white pulp, central artery is called artery of pulp, which branch into small penicillate arteries. Their branches are called encapsulated arterioles because they are surrounded by many phagocytosing cells. Encapsulated arterioles branch into precapillary arterioles. There are two theories explaining how the blood from these precapillary arterioles drain into venous sinuses. First theory, theory of closed circulation, tells that the blood from arterioles flows directly into venous sinuses. Second theory of open circulation explains that this blood is at first poured into cords of Billroth and then drained into venous sinuses. Probably there's a combination of both of these processes. Spleen plays a number of essential functions in the human body. It's a member of the body's immune defense, it serves as a blood filter, it destroys and absorbs dying damaged erythrocytes, leukocytes, and bacteria. During the fetal period, hemopoiesis occurs here. And yeah, that's all you need to know about basics of spleen histology. Thanks for watching. In memory of Professor Ludek Weiner, who has been the head of the Institute of Histology and Embryology of the Second Faculty of Medicine in Charles University since 2003, and has made a significant contribution to the quality of teaching and functioning of our institute. Thank you.